vaccine, not Astra. Bharat Biotech.
India में तो
That's adenovirus, yeah?
No, that is attenuated virus.
Oh, okay.
But 80% population has the AstraZeneca.
Yeah, yeah. Made in India, though.
Made in India. Yeah.
Yeah, yeah.
But Covaxin is also made in India.
Ah, okay.
I don't know whether you remember Bharat Biotech. There is a company, Chetan was collaborating.
Yeah.
So Sanjay is making mRNA vaccine.
Yeah. I haven't seen Sanjay for a while.
Yeah, he gave the first talk yesterday.
Has he got an mRNA malaria vaccine yet?
He said he's trying.
Yeah.
Multi-stage. So, is he making your RH5?
I don't know. He'd be crazy if he wasn't.
Yeah, we should have discussed with him. Whether he's making. Yeah. It's a very good candidate.
Yeah. Not on its own. Yeah. So multi-stage is a good way to go.
Yeah.
How's ICGB going?
It's fine. We lost a lot of good people, but still we are managing.
अनम्यूट करो।
Alan? Hello? Alan?
Yeah.
Should we start?
Sure.
It gives me...
Recording in progress.
Okay, welcome to the second day of the discovery of novel anti-malarial and drug and vaccine targets in malaria and TB. And it gives me a great pleasure to introduce Professor Alan Coleman, who has been a great friend of ICGB. And he is one of the leading malaria scientists and has contributed a lot in the basic malaria research as well as the translational research. In fact, many of us has been following his work very minutely, and he has been mentor of most of us at ICGB as well as many Indian scientists. We always have learned a lot from Alan, and he has visited us a number of times. In fact, he organized one of the first transfection workshop at ICGB New Delhi, where a number of students as well as scientists from all over the world came and learned a number of technologies, genetic manipulation technologies from Alan and his group. In fact, his whole group visited us and taught us a number of nice transfection technology as well as genetic manipulations. His main contribution at ICGB in the world in malaria biology has been that how parasite invade RBC, and he has been able to really identify many novel host-parasite interactions. And I really have learned a lot from him, and he is a great friend of all of us, and never said no to us whenever we invited him to deliver a talk. And we are very grateful to Alan. And now I give the dice to Alan to please give us a talk.
Thank you very much, Pawan, and thank you very much to you and ICGEB for the invitation to speak at this workshop. I'd certainly be much happier to be in Delhi, but it's much cooler in Melbourne at the moment. So let me tell you about some of the work that we've been doing in a collaboration with Merck in the USA, as well as Wuxi in China. And it's with respect to the discovery of novel anti-malarials. And we've been using them as chemical tools to really discover new biology of the malaria parasite. And let me just talk firstly about the issue with respect to drug resistance in Plasmodium falciparum. And Plasmodium falciparum has become resistant to many of the anti-malarials that are used against it, but also, very concerningly, increased resistance to artemisinin and its partner drugs. What I'd like to show you here is essentially world maps and the emergence of resistance or treatment failures to artemether and lumefantrine here. And as you can see, as time progresses, you see spread of that throughout the world through to Africa. On the bottom, you see the prevalence of molecular markers of artemisinin resistance, and again, through time, and you see those molecular markers spreading from Southeast Asia across India and to Africa. So one of the risks of this is certainly that we will run out of potential good treatments and prophylactic controls for Plasmodium falciparum and also other Plasmodium species that cause malaria in humans. So we really need to have a robust pipeline for the development of new antimalarials. And we need novel molecules intended for that must not be cross-resistant with the existing therapies. And so that's one of the aims that we started with when we started a collaboration with Merck about 5 or 6 years ago, when they contacted us and asked whether we were interested in collaborating to try and develop new antimalarial drugs. And so what we decided to do was, with Merck, to screen a library of compounds that they had that targeted aspartic proteases, particularly in humans, but also other pathogens of humans, and to essentially screen them against Plasmodium falciparum in a phenotypic assay to try and identify those that stop the parasite from developing in the blood stage of the parasite. And with this screen, we identified one drug-like hit class, of which there were 32 different examples. And this just shows a determination of EC50 of one of those compounds, which I've called WM4. And in this case, we've tested the two different enantiomers of WM4, the R enantiomer and S enantiomer, just to show that, in particular, the R enantiomer is the active component of this compound that we identified in this high-throughput screen. So essentially, we developed a nice hit class of drug-like compounds that we hoped we could develop into drug compounds that could be developed further into antimalarial drugs. So what firstly was the target of those compounds? And initially we didn't know what it was. We thought it was probably an aspartic protease. We're actually hoping it was an aspartic protease of Plasmodium falciparum called plasmepsin 5, but it turned out not to be that. It was actually targeting plasmepsin 10. And we showed that by a number of methods, including selection of drug-resistant P. falciparum parasites in vitro, where we could select over about a 6 to 8 month period resistance to WM4 that was about 14 to 16-fold more resistant. We then sequenced the genome of Plasmodium falciparum and showed that in those selected parasites, there was an amplification event. And within that amplification event, there was a gene that encoded plasmepsin 10, which is an aspartic protease. We showed that in those resistant parasites, you get overexpression of plasmepsin 10, as you'd expect, because of the increased gene copy number. But we also showed by a number of other ways that these compounds were actually targeting plasmepsin 10. We showed that by synthesizing, firstly, a cross-linking analog of these compounds and immunoprecipitating, and also by making gene knockdowns of plasmepsin 10 and showing that they become more sensitive to these compounds. So therefore showing them that that is indeed the target. So WM4 specifically targets the aspartic protease plasmepsin 10. So I'm not telling you the story in the direct chronology of how we did the experiments. It's a bit easier to jump ahead, and it certainly took a while to express plasmepsin 10, but we also recombinantly expressed other plasmepsins, including the P-Vivax plasmepsin 10, P-Falciparum plasmepsin 10, as well as P-Falciparum plasmepsin 9, which I'll talk quite a bit about as well. And what you can see here is that recombinant expression and then purification of each of these proteases. We developed assays for each of these proteases because we had new different protein substrates of these proteases and the particular peptide that they cleaved, and so we could develop this FRET-based assay that enabled us to essentially measure activity. And once you have that, then you can do Mercatos Kinetics, and what we wanted to show or determine was the inhibitory constant for WM4 with respect to P-Falciparum plasmepsin 10, P-Vivax plasmepsin 10, but also P-Falciparum plasmepsin 9. And this is the EC50, the concentration that kills 50% of parasites for WM4, so it's 7.5 nanomolar. So it's a very potent compound, and very potent for a first hit of a drug-like compound that we'd identified. If you look at the Ki, the inhibitory constant for P-Falciparum plasmepsin 10, you see it's 0.4 nanomolar. So again, very potent inhibitor, and basically shows that it is indeed the target for this drug. This protease is the target. P-Vivax plasmepsin 10, the Ki is again about 0.4 nanomolar, very potent. But you see for plasmepsin 9, these compounds are nowhere near as potent, and the fold selectivity for the WM4 for plasmepsin 10 over plasmepsin 9 is about 590 fold. So it's targeting plasmepsin 10, and that is the reason it kills Plasmodium falciparum in vitro. So it's a specific inhibitor. It's a good hit compound for medicinal chemistry program to develop lead compounds, which is what we set about doing in collaboration with Merck. And this essentially is the scheme that we followed over a couple of years to develop lead compounds and then clinical candidates for this class of compounds, where essentially we were designing compounds based on primary assays against the parasite, as well as secondary assays looking at enzyme activity, as well as the inhibition of the function of the protein, of the drugs in P. falciparum, as well as their efficacy in mouse models of malaria, their bioavailability, toxicity, and metabolism. And then using this information to design new compounds using structural information. I'll show you some of that as well. Compound design, and then chemical synthesis of these design compounds with WuXi Abtec, and these compounds come back to WeHi and then goes around this circle. And we'd essentially synthesized and designed more than 4,000 of these compounds through this program, and eventually identified this compound that we've called WM382, which is our lead compound. And I'll talk quite a bit about this compound as well as WM4. I'll mention a little bit about a compound which I'll call WM06. Sorry, it keeps jumping ahead a bit. WM06, and I'll come back to that. And this is our advanced compound that is moving towards human clinical trials. So WM382 was interesting because as we were developing it, we realized something really important. It was very potent, and this just shows you the EC50 against P. falciparum in vitro. It's 0.39 nanomolar, so quite a bit more potent than WM4. And that potency, as I'll show you, is because of the fact that it hits two targets. It hits plasmepsin 10, but it also hits plasmepsin 9. And this dual selectivity or specificity for plasmepsin 10 and plasmepsin 9 is really important in terms of its potency, but also, as I'll show you later, for its ability to prevent development of resistance. So if you look at the Mercatos Kinetics, firstly WM4, I've shown you this data. WM382, essentially you see that it's the Ki is quite a bit more potent, 0.014 nanomolar, and for P-Vivax plasmepsin 10, 0.007 nanomolar, and for Plasmodium Falciparum plasmepsin 9, 0.4 nanomolar, as compared to WM4, which is 263 nanomolar. So the false selectivity for WM382 of plasmepsin 10 over 9 is 29-fold. So this compound is hitting both plasmepsin 10 and plasmepsin 9. So we have a dual aspartic acid protease inhibitor, as opposed to WM4, one of our initial hits, which only really hits plasmepsin 10. So WM4, and I haven't mentioned WO5, but they are essentially specific inhibitors of plasmepsin 10. WM382 is a dual inhibitor of plasmepsin 10 and 9, and they really gave us chemical tools to understand the function of plasmepsin 9 and 10, because we had WM4, a specific inhibitor of plasmepsin 10, and WM382 that inhibited 10 and 9. And as Parwin mentioned, we work quite a bit on invasion of P. falciparum into human red blood cells. And these two projects really came together when we started to realize and show that plasmepsin 9 and 10 really are responsible for processing some of the important proteins involved in the invasion process that we are working on, namely the RH proteins and the EBA proteins, and EBA for erythrocyte binding antigens and RH for reticulocyte binding like homologs. These bind to particular receptors on the red blood cell. And one complex we're particularly interested in is this RH5 complex, which is also an RH protein, but quite different to the other ones. It forms a complex with RIPA and SERPA or CYRPA. So we were interested in the function of these proteins and really started to understand that these proteases, plasmepsin 9 and 10, are really important in terms of processing of these ligands and important for their function in terms of binding to receptors on the red blood cell surface. So we could show that by a number of ways. So one was to essentially synthesize peptides, FRET-based peptides, where we could assay for cleavage of those sequences by plasmepsin 10 from Falciparum, plasmepsin 10 from Vivax, but also plasmepsin 9. And that's what we've done here, where we've essentially shown that peptides to specific areas of the protein that we'd identified as potential targets for proteolysis actually are cleaved by plasmepsin 10, and the substrate specificity essentially identical for P-Vivax plasmepsin 10, but somewhat different for plasmepsin 9. Although plasmepsin 9 could cleave what we were plasmepsin 10 substrates, and plasmepsin 10 could also cleave plasmepsin 9 substrates. So they really did share some substrate specificity, but there were differences as well in terms of that substrate specificity. And I won't go through this in detail, but this is essentially looking at some of those substrates that we and others had identified, Dominic Soldati's lab and Dan Goldberg's lab, and the sequences that they cleaved. And we showed this using a number of methods, including pull downs and proteomics. So this is just a summary of the cleavage recognition site, and the plasmepsin 10 and plasmepsin 9 cleave after this P1 amino acid here, which is predominantly phenylalanine in most of the cases. So what we did show, following on work again from Soldati and Goldberg lab, was that plasmepsin 9 and 10 are master regulators of both egress of P. falciparum from the red blood cell, but also invasion of human red blood cells. So I'm not going to show you a lot of data showing that. What I'd like to show is a summary of a lot of that data. And first thing that we showed is plasmepsin 9 and plasmepsin 10 are autocatalytic. They activate themselves. And that WM382 inhibits both plasmepsin 9 and 10 because it's a dual inhibitor, whereas WM4 is more selective with respect to plasmepsin 10 inhibition. So we could show that both of those compounds inhibit processing of these RH proteins, including RH5, as well as these EVA proteins, including 175, 140, and 181, which bind to these particular receptors. They also, this protease also is responsible for processing really important proteins in the invasion process, including apical membrane antigen 1, as well as these other ones, RIPA, which forms the complex with RH5, PTRAMP, which is important in invasion. We also, sorry about that, showed that plasmepsin 10, sorry, I have it set on a timer apparently. Plasmepsin 10 activates subtilisin 2 and is responsible for processing AMA1, MSPB1 and PTRAMP and other proteins. Subtilisin 1, which we already knew from Goldberg and Soldati labs, was responsible for, essentially was processed by plasmepsin 10, and it is responsible for cleaving quite a few proteins that are important for invasion and other processes. Plasmepsin 9, autocatalytic, and WM382 inhibits it, but it's also responsible for processing proteins such as apical sushi protein, not RIPA, incorrect here, RAP1, RON3, MTRAP, but also other proteins that we've identified fairly recently, RON6, RAMA, and RELP1. Is there more? Certainly there are more substrates. These proteases seem to process quite a lot of different proteins. So why is WM382 so potent, and why and how does it have dual activity against plasmepsin 9 and 10? So we moved to surface plasmon resonance to look at essentially the affinity of binding of different compounds to these different aspartic proteases. And firstly, WM4 is just showing some SPR traces here, and working out the affinity of binding, and the KD is 0.48 nanomolar, so really quite tight binding, but you can get a measurable off rate for this compound. And if you compare it to plasmepsin 9, and you see the KD is certainly much higher, so the affinity of binding is much less, as you would expect, because the specificity for WM4 is as a plasmepsin 10 inhibitor, and is much worse as a plasmepsin 9 inhibitor. But you look at WM382, and firstly with Plasmodium bivax plasmepsin 10, and you see a fast on rate and a very, very slow off rate. In fact, it's not measurable, and it's not measurable, it's not possible to measure a KD. We can only estimate it at less than 0.1 nanomolar. So essentially, these compounds bind and have an extremely slow off rate. If we look at the plasmepsin 9, we see exactly the same thing for WM382, which, as I've told you, is a dual inhibitor against both of these aspartic proteases. And again, a very quick on rate, and the off rate is essentially not measurable, and so we can't measure a KD for that. So we know it binds and essentially doesn't come off. So really an excellent inhibitor of both of these proteases. So the potency of WM382 is against Plasmodium falciparum is because it's a dual inhibitor of both plasmepsin 9 and 10, and has an extremely slow off rate for binding. So how does it bind, and can we work out why 9 is essentially a good, WM382 is a good inhibitor of both 9 and 10 aspartic proteases, whereas WM4 is selective for plasmepsin 10. And to do that, we moved to structural biology, and here we've worked out the structure of the plasmepsin 10, Falciparum plasmepsin 10 enzyme at 1.8 angstroms. And what I've shown here are the two aspartic catalytic amino acids in the active site. And you see this nice flap closing over it, which is very typical of these aspartic proteases. So if we move to essentially looking at the structure of the compounds in the active site, and we did this by solving crystal structures of P-Falciparum plasmepsin 10 with WM382, as well as P-Vivax plasmepsin 10 with WM382 and WM4. What I've shown here is another compound I've called WM06, and I won't show any of the structures of that. That is one of our advanced compounds that we've also sold structures in complex with plasmepsin 10. And I won't go into this in a lot of detail, but what it enabled us to do was essentially to work out the molecular interactions of WM4 and WM382 in the active site of plasmepsin 10, and really understand why WM4 binds with less affinity compared to WM382, and use the plasmepsin 9 model structure to work out why WM4 essentially is not able to inhibit it, but WM382 is. Firstly, what we've shown is WM4 has interactions with the S1' and S2' substrate binding pockets, which is really important for its affinity of binding to plasmepsin 10. But when you look at WM382, you see those similar interactions, but you also see really important extra interactions with the flap. And essentially, the flap region is this region here that is closing over WM382 that is sitting in this active site. And you see interactions of WM382 with this flap region, particularly serine in that flap region. And that's really important with respect to plasmepsin 9, because you still have the same flap region, but you don't have a serine. Instead, you have a threonine, which is not able to form that hydrogen bond. And so that's one of the reasons why WM382 is essentially a dual inhibitor. It has extra interactions in the active site, whereas WM4 also has those interactions, but not those extra interactions with the flap of plasmepsin 9, at least in the model structure. We as yet do not have the plasmepsin 9 crystal structure. So what is the mode of action of these compounds? How do they block parasite growth? And what I've shown here is essentially a time course showing that WM382 essentially blocks the late stage, the Schizon stage of Plasmodium falciparum. Essentially, it blocks egress, and you can show that quite clearly, where I'm comparing WM4 and WM382. Similar inhibition at egress. You do not get egress of the merozoites from Schizons. And you can see that quite nicely by these movies. And on the left is wild-type parasites, and what you're seeing is egress of merozoites out of the red blood cell. Whereas WM382 treated parasites, you essentially see dead parasites. You see no egress of those parasites out of red blood cells. If you break those merozoites out of red blood cells and ask, can they invade red blood cells? The answer is no. So they are not able to invade merozoites. And we believe it's because a lot of the proteins required for that invasion process are not correctly processed due to inhibition of plasmepsin 10 in particular, but also plasmepsin 9. What happens in vivo, and essentially WM382 is orally available. It can cure mice of both P. burgii, but also mouse models of P. falciparum infection. This is the P. burgii, one of the P. burgii experiments where we show that WM382 at 60 and 30 milligrams per kilogram will cure infection of Plasmodium burgii. Also, if you use P. falciparum models of humanized models of P. falciparum, what we're comparing chloroquine at 50 milligrams per kilogram and WM382 at 1, 3, 10, and 30 milligrams per kilogram. You see, firstly, that WM382 at 1 milligram per kilogram does not cure or does not suppress growth of the parasite, whereas 3, 10, and 30 does, and suppresses as well as chloroquine at 50 milligrams per kilogram. In the case of the higher dose, is faster in that suppression. So WM382 is superior to chloroquine in its ability to cure mice with P. burgii and P. falciparum infection. What about liver stages of P. burgii infection? And in this case, we're infecting mice either with sporozoites or with mosquito bites that have been infected with P. burgii parasites, and then visualizing those parasites essentially through luciferase. And what you see is infection in the liver, in hepatocytes, when there's no treatment, but also with treatment with WM382 at 2 different doses, 100 milligrams per kilogram and 20 milligrams per kilogram. You see as these parasites get released from the hepatocytes, they move into the blood circulation of the mouse, but not for WM382 with both of these doses. So WM382 given orally to mice blocks movement of P. falciparum from liver stage to blood stage infection. And you can quantitate that and show that quite well here with 2 doses of 100 milligrams per kilogram or 1 dose of 100 milligrams per kilogram in these examples. So a single dose cure of mice with P. burgii infection when given WM382 orally at liver stage. So what about transmission? And this is P. falciparum, and you can show similar experiments with P. burgii, but this is the in vitro experiments where we fed gametocytes to P. falciparum, treated with WM382 in this case, and shown that this blocks oocyst formation in the mosquito gut. And this is while non-treated gut, where we're counting the oocysts in the gut, and this is a gut that's been treated with WM382, or mosquitoes, gametocytes that have been treated with WM382. This is quantitation here showing the difference between WM382 and WM4, which was nowhere near as good. So it suggested to us that Plasmodium 10 was perhaps not as important in gametocytogenesis as perhaps Plasmodium 9. So what we've shown is that WM382 and the derivatives that we've since developed from WM382 are excellent in terms of their potential as potential anti-malarial drugs, because they will block liver stage development of P. burgii and P. falciparum, or P. burgii, as we've shown. They'll block blood stage development by blocking both Plasmodium 9 and 10, and they will block transmission to mosquitoes. So an important aspect which I started this talk with is that we really need to develop new anti-malarials that certainly aren't cross-resistant with already used current anti-malarial drugs such as artemisinin and its derivatives, but also other drugs such as chloroquine and other anti-malarials. And so what we did was determine whether there was any cross-resistance with respect to the compounds we developed that were hitting Plasmodium 9 and 10 in parasites that were resistant to other anti-malarial drugs. And this is a summary. I haven't put the numbers here because it's just easier to look at in colours. Green says essentially there is no resistance. And so these are different P. falciparum lines that are resistant to different anti-malarial drugs. In this case, it's artemisinin, mefloquine, atovaquone, and other compounds that are currently in clinical trials, chloroquine and mefloquine for these other compounds. And red means that they're resistant. Sorry about that. resistant, whereas green not resistant. And what you see over here is WM382. You see absolutely no cross-resistance with any of these parasites, as is this more advanced drug, which I've called WM06. Again, no cross-resistance. And we certainly want to know whether it was possible to develop resistance to WM382 and other compounds such as WM06. And we did this essentially by a number of different ways, but in particular by time to resistance studies, where we use different inoculums of parasites and try to develop resistance with essentially a reasonably large selective pressure of drug, in this case about the IC90-95 or EC90-95 for the compound. So essentially we're looking for the development of resistance compared to known antimalarials such as the tovaquone. And what you see here is selective pressure and development of resistance or resistance acquisition, and then the number of days essentially that resistance developed. And generally it's between 30 to 60 days for a tovaquone. For Genoplaside, again, we could develop resistance, and again, it's a similar time frame. For WM4, although we could develop resistance if we slowly selected resistance over a long period of time, in these time to resistance studies, we could not develop resistance. WM382, we could not develop resistance no matter how we tried, and that was also true of WM06. So what I've told you about is work that we've been doing with Merck to identify a novel class of compounds that are potent inhibitors of P. falciparum. We know they inhibit P. knowlesi and P. berghii. We also expect that they inhibit other Plasmodium species, including Plasmodium bivax, because we've shown they essentially, these inhibitors target the plasmepsin 9 and 10 proteases that are very conserved. We've used structure-guided medicinal chemistry to develop novel lead compounds and essentially move those into clinical development. We know that these are dual-targeting compounds that target both plasmepsin 9 and 10. And once we knew that, that was really important in terms of their potency, as well as the difficulty in the ability of the parasite to develop resistance, we made sure we maintained that selectivity on both 9 and 10 throughout our drug development program. We've done in vivo proof of concept by oral dosing of these dual inhibitors, and shown that you can get full efficacy and cure of P. berghii as well as humanized mice with P. falciparum. We've shown that you cannot select you do not get cross-resistance to current antimalarials. Perhaps not unexpected, because they have a novel mechanism of action. There's a very high bar for selection of drug resistance, and suggests that they are in a class of what we would call irresistible. We've shown that these compounds disrupt the 3 stages of the Plasmodium life cycle: liver, blood, and transmission stage. And they really are candidates for MMB target product profiles of TPP1 and TPP2, in other words, for treating, as well as for chemo protection. We've used these as chemical tools to really understand the function of plasmepsin 9 and 10. And I haven't shown you all the data with respect to that. I didn't want to get into the minutiae of all of the proteins that they're processing and how they do that. But they're certainly master regulators of parasite egress, invasion, and development by being required for processing, essential processing of important proteins in those processes. And these programs are certainly not done by a few people. Really large teams are absolutely essential for these programs, both for the drug development, but also for the biology that we've done with these different compounds. Teams at WEHI, and I won't go through all the names here, but also at Merck. And I'd like to mention David Olson in particular, who heads the team at Merck, as well as Manuel de Lira Ruz, who heads the medicinal chemistry team at Merck as well. We did this in collaboration with Wuxi, who basically were responsible for synthesizing a lot of the more than 4,000 compounds that we've synthesized through this program. And this was funded by Wellcome Trust, and I'd like to mention our advisors from Wellcome Trust, Rick Keenan and Jens Keckbush. Thank you very much.
Thanks, Alan. Really, we are, every time whenever we listen to you, we always listen to you something new and novel. It's fantastic. I think this inhibitor should be in the clinical trial next few months. You have a few questions. If you permit, we can ask you.
Yep.
So, Alan, why these compounds don't act on other aspartic proteases because of some of the loops which you have shown, or because do you see any food vacuole abnormality or other plasminogen 1, 2, or 4?
Yeah. We certainly see no activity on plasminogen 5 at all. And during the medicinal chemistry program, we screened human aspartic proteases as well, including base renin and others, to make sure that we weren't targeting the human aspartic proteases and that we were being selective with respect to Plasmodium aspartic proteases. In terms of plasmepsin 1, 2, 3, 4, 7, 8, we haven't specifically expressed those, but we have we have made compounds that are cross-linkable and then done pull downs, and we don't pull down 1 to 4, plasmepsin 1 to 4, or plasmepsin 5, or plasmepsin 7 or 8. If we select resistance with using WM4, which is targeting only plasmepsin 10, so we can do that over a long time. We essentially only amplify plasmepsin 10, so it suggests that it's only hitting plasmepsin 10. We see no changes in any other plasmepsin genes. So we really think that these compounds are very selective for plasmepsin 9 and 10, and we've ensured that right throughout the medicinal chemistry program.
Okay, Pushkar has one question. Pushkar, please go ahead.
Hi, Alan. Good to see you after long.
Hi, thank you. Good to see you.
Good to see you. Just a quick question. Do you think 9 and 10, if they work independently or they work in tandem?
Yeah, they definitely work. Well, I believe they work independently. Plasmepsin 10 is located in micronemes. We think plasmepsin 9 is predominantly in the neck of the rhopteries, although it could start in a subclass of micronemes that is different to plasmepsin 10, but it appears mostly in the neck of the rhopteries. We are pretty sure that from all the substrates we have identified, and others from Soldati and Goldberg, that 9 processes mostly rhoptery proteins, whereas plasmepsin 10 processes mostly micronemal proteins. They probably do end up on the surface of the merozoite, and they might mix together at that stage, but they have a pH activity in the low pH 6, pH 5 range. So they need an acidic environment for good activity. So once they end up in pH 7, which is what the pH would be once they end up on the outside of the merozoite, they're probably inactive. So they're active in a acidic pH environment. The micronemes are certainly acidic, more acidic. Rhoptery neck, we don't know, but we'd expect that that's probably going to be somewhat acidic as well. So their activity is probably controlled by that pH of the particular organelles as well. So we think they're operating totally independently.
If I may, one quick question. Is sub-1 by any chance a putative or a real substrate for any of these?
Yeah, absolutely. Sub-1 is the substrate for plasmepsin 10. It probably gets processed in micronemes and then put onto the surface of the merozoite. If it doesn't get cleaved by plasmepsin 10, it's not active, so it's an essential step. Subtilisin 2 is also a substrate of plasmodioxin 10, and again, if it doesn't get processed, it's not activated. So plasmodioxin 10 is really important for the proteolytic activation of both subtilisin 1 and subtilisin 2.
Thanks, Alan.
Thank you.
Someone from CDRI also would like to ask you.
Yes, Professor Common, a lovely talk, and thanks very much for the for the update on the molecules. A quick question: what's the PK like for this molecule? Is it a long-lasting one, or do you see active metabolites as well? And would you then position this as a single-dose molecule, or are you planning to give it orally for 2 or 3 days?
Yeah, I didn't show any of the pharmacokinetics or pharmacodynamics that has been developed. That's another seminar in its own right. And it depends which compound you talk about. So WM382, even though I've shown you in vivo data and most of the data with respect to showing that it's a potential anti-malarial, is never going to be an anti-malarial because its pharmacokinetics are not good enough, although it works really well in mice, no doubt about that. That was essentially one of our lead compounds. We've gone a lot further now, and we've developed very advanced lead compounds that have a much longer half-life and much better pharmacokinetics and safety profiles in terms of their activity in dogs and rats. And so that program has been going on for quite a while, and we're essentially moving into clinical trials in the near future. So WM382, no, absolutely not. It's not a new anti-malarial, but the compounds that we've developed since then certainly are looking very good.
Thank you.
A student from Delhi University has asked a question: do these compounds act on cerebral malaria, severe malaria?
Hello?
Hello?
Yeah, I'm still there.
So a student had asked a question on chat box that do these compounds act on severe malaria or cerebral malaria?
Oh, sorry. I thought the student was going to ask it. We would certainly expect so. We haven't tried it in any of those models, so I can't answer the question specifically. These compounds, if in vitro, they're medium fast in terms of their activity. In vivo, in mice, they're certainly fast in terms of their anti-malarial activity. So they're essentially acting from troph to chizont and medium fast. So we would expect them to be quite good in terms of affecting parasites that are about to cytoadhere or to adhere. But specifically, we haven't looked at that.
One last question. How does this compound block the egress? Because they are acting on most of the merozoite surface molecules. Do they also act on cytoskeleton, RBC cytoskeleton proteins or parasite cytoskeleton proteins?
Yeah, a great question, and we don't know the answer to that one. Certainly, we're in the process of identifying all the substrates of plasmodioxin 9 and 10 using proteomic approaches. We know a lot, and I showed you a summary of the ones that we do know currently, and we certainly think that the egress inhibition is by essentially blocking processing of proteins that are essential for egress, rather than them acting directly onto cytoskeleton of the red blood cell. So, but it's something that we're certainly interested in looking at, but we haven't specifically done it yet.
Thanks, Alan. It's a great talk.
Thank you.
And we always learn something new from you. Always. That's why you have been our mentor for a long time. Thanks a lot.
Thank you very much. Thanks, Pawan. Thank you.
And you can stop sharing, please?
Yeah, I'm just working out how to do it. Sorry. Yeah, stop share. There we are. Okay.